10.3 Microevolution and the Genetics of Populations. Lesson Objectives. Distinguish between microevolution and macroevolution. Define gene pool, and explain how to calculate allele frequencies. State the Hardy-Weinberg theorem. Identify the four forces of evolution. Vocabulary. Allele frequency. Directional selection. Disruptive selection. Gene flow. Gene pool. Genetic drift. Hardy-Weinberg theorem. Macroevolution. Microevolution. Population genetics. Sexual dimorphism. Stabilizing selection. Introduction. Darwin knew that heritable variations are needed for evolution to occur. However, he knew nothing about Mendel's laws of genetics. Mendel's laws were rediscovered in the early 1900s. Only then could scientists fully understand the process of evolution. The scale of evolution. We now know that variations of traits are heritable. These variations are determined by different alleles. We also know that evolution is due to a change in alleles over time. How long a time? That depends on the scale of evolution. Microevolution occurs over a relatively short period of time within a population or species. The Grants observed this level of evolution in Darwin's finches. Macroevolution occurs over geologic time above the level of the species. The fossil record reflects this level of evolution. It results from microevolution taking place over many generations. Genes in populations. Individuals do not evolve. Their genes do not change over time. The unit of evolution is the population. A population consists of organisms of the same species that live in the same area. In terms of evolution, the population is assumed to be a relatively closed group. This means that most mating takes place within the population. The science that focuses on evolution within populations is population genetics. It is a combination of evolutionary theory and Mendelian genetics. Gene pool. The genetic makeup of an individual is the individual's genotype. A population consists of many genotypes. Altogether, they make up the population's gene pool. The gene pool consists of all the genes of all the members of the population. For each gene, the gene pool includes all the different alleles for the gene that exist in the population. For a given gene, the population is characterized by the frequency of the different alleles in the gene pool. Allele frequencies. Allele frequency is how often an allele occurs in a gene pool relative to the other alleles for that gene. Look at the example in table below. The population in the table has 100 members. In a sexually reproducing species, each member of the population has 2 copies of each gene. Therefore, the total number of copies of each gene in the gene pool is 200. The gene in the example exists in the gene pool in 2 forms, alleles A and A. Knowing the genotypes of each population member, we can count the number of alleles of each type in the gene pool. The table shows how this is done. Genotype. Number of individuals in the population with that genotype. Number of allele A contributed to the gene pool by that genotype. Number of allele A contributed to the gene pool by that genotype. AA. 50. 50 times 2 equals 100. 50 times 0 equals 0. AA. 40. 40 times 1 equals 40. 40 times 1 equals 40. AA. 10. 10 times 0 equals 0. 10 times 2 equals 20. Totals. 100. 140. 60. Let the letter P stand for the frequency of allele A. Let the letter Q stand for the frequency of allele A. We can calculate P and Q as follows. P equals number of A alleles / total number of alleles equals 140 / 200 equals 0.7. Q equals number of A alleles / total number of alleles equals 60 / 200 equals 0.3. Notice that P plus Q equals 1. Evolution occurs in a population when allele frequencies change over time. What causes allele frequencies to change? That question was answered by Godfrey Hardy and Wilhelm Weinberg in 1908. The Hardy-Weinberg theorem. Hardy was an English mathematician. Weinberg was a German doctor. Each worked alone to come up with the founding principle of population genetics. Today, that principle is called the Hardy-Weinberg theorem. It shows that allele frequencies do not change in a population if certain conditions are met. Such a population is said to be in Hardy-Weinberg equilibrium. The conditions for equilibrium are: no new mutations are occurring. Therefore, no new alleles are being created. There is no migration. In other words, no one is moving into or out of the population. The population is very large. Mating is at random in the population. This means that individuals do not choose mates based on genotype. There is no natural selection. Thus, all members of the population have an equal chance of reproducing and passing their genes to the next generation. When all these conditions are met, allele frequencies stay the same. Genotype frequencies also remain constant. In addition, genotype frequencies can be expressed in terms of allele frequencies, as table below shows. Genotype. Genotype frequency: AA, P2, AA, 2PQ, AA, Q2. Hardy and Weinberg used mathematics to describe an equilibrium population: P equals frequency of A, Q equals frequency of A, P2 plus 2PQ plus Q2 equals 1. In table above, if P equals 0.4, what is the frequency of the AA genotype? A video explanation of the Hardy-Weinberg model can be viewed at 1457. Forces of evolution. The conditions for Hardy-Weinberg equilibrium are unlikely to be met in real populations. The Hardy-Weinberg theorem also describes populations in which allele frequencies are not changing. By definition, such populations are not evolving. How does the theorem help us understand evolution in the real world? From the theorem, we can infer factors that cause allele frequencies to change. These factors are the forces of evolution. There are 4 such forces: mutation, gene flow, genetic drift, and natural selection. Mutation. Mutation creates new genetic variation in a gene pool. It is how all new alleles first arise. In sexually reproducing species, the mutations that matter for evolution are those that occur in gametes. Only these mutations can be passed to offspring. For any given gene, the chance of a mutation occurring in a given gamete is very low. Thus, mutations alone do not have much effect on allele frequencies. However, mutations provide the genetic variation needed for other forces of evolution to act. Gene flow. Gene flow occurs when individuals move into or out of a population. If the rate of migration is high, this can have a significant effect on allele frequencies. Both the population they leave and the population they enter may change. During the Vietnam War in the 1960s and 1970s, many American servicemen had children with Vietnamese women. Most of the servicemen returned to the United States after the war. However, they left copies of their genes behind in their offspring. In this way, they changed the allele frequencies in the Vietnamese gene pool. Was the gene pool of the American population also affected? Why or why not? Genetic drift. Genetic drift is a random change in allele frequencies that occurs in a small population. When a small number of parents produce just a few offspring, allele frequencies in the offspring may differ, by chance, from allele frequencies in the parents. This is like tossing a coin. If you toss a coin just a few times, you may, by chance, get more or less than the expected 50% heads or tails. In a small population, you may also, by chance, get different allele frequencies than expected in the next generation. In this way, allele frequencies may drift over time. There are 2 special conditions under which genetic drift occurs. They are called bottleneck effect and founder effect. Bottleneck effect occurs when a population suddenly gets much smaller. This might happen because of a natural disaster such as a forest fire. By chance, allele frequencies of the survivors may be different from those of the original population. Founder effect occurs when a few individuals start, or found, a new population. By chance, allele frequencies of the founders may be different from allele frequencies of the population they left. An example is described in figure below. Figure 1. Founder effect in the Amish population. The Amish population in the US and Canada had a small number of founders. How has this affected the Amish gene pool? Natural selection. Natural selection occurs when there are differences in fitness among members of a population. As a result, some individuals pass more genes to the next generation. This causes allele frequencies to change. The example of sickle cell anemia is described in figure below and table below. It shows how natural selection can keep a harmful allele in a gene pool. You can also watch a video about natural selection and sickle cell anemia at this link. Figure 2. Sickle cell and natural selection. Genotype. Phenotype. Fitness. AA. 100% normal hemoglobin. Somewhat reduced fitness because of no resistance to malaria. As. Enough normal hemoglobin to prevent sickle cell anemia. Highest fitness because of resistance to malaria. SS. 100% abnormal hemoglobin, causing sickle cell anemia. Greatly reduced fitness because of sickle cell anemia. Here's how natural selection can keep a harmful allele in a gene pool. The allele, S, for sickle cell anemia is a harmful autosomal recessive. It is caused by a mutation in the normal allele, A, for hemoglobin, a protein on red blood cells. Malaria is a deadly tropical disease. It is common in many African populations. Heterozygotes, as, with the sickle cell allele are resistant to malaria. Therefore, they are more likely to survive and reproduce. This keeps the S allele in the gene pool. The sickle cell example shows that fitness depends on phenotypes. It also shows that fitness may depend on the environment. What do you think might happen if malaria was eliminated in an African population with a relatively high frequency of the S allele? How might the fitness of the different genotypes change? How might this affect the frequency of the S allele? Sickle cell trait is controlled by a single gene. Natural selection for polygenic traits is more complex, unless you just look at phenotypes. 3 ways that natural selection can affect phenotypes are shown in figure below. You can also watch an animation comparing the 3 ways at the link below. Stabilizing selection occurs when phenotypes at both extremes of the phenotypic distribution are selected against. This narrows the range of variation. An example is human birth weight. Babies that are very large or very small at birth are less likely to survive. This keeps birth weight within a relatively narrow range. Directional selection occurs when one of 2 extreme phenotypes is selected for. This shifts the distribution toward that extreme. This is the type of natural selection that the Grants observed in the beak size of Galapagos finches. Disruptive selection occurs when phenotypes in the middle of the range are selected against. This results in 2 overlapping phenotypes, one at each end of the distribution. An example is sexual dimorphism. This refers to differences between the phenotypes of males and females of the same species. In humans, for example, males and females have different heights and body shapes. Figure 3. Natural selection for a polygenic trait. Natural selection may affect the distribution of a polygenic trait. These graphs show 3 ways this can happen. For a review of natural selection and genetic drift, and how they relate to evolution, see mutation, natural selection, genetic drift, and gene flow are discussed at 8:45. Lesson summary. Microevolution occurs over a short period of time in a population or species. Macroevolution occurs over geologic time above the level of the species. The population is the unit of evolution. A population's gene pool consists of all the genes of all the members of the population. For a given gene, the population is characterized by the frequency of different alleles in the gene pool. The Hardy-Weinberg theorem states that, if a population meets certain conditions, it will be in equilibrium. In an equilibrium population, allele and genotype frequencies do not change over time. The conditions that must be met are no mutation, no migration, very large population size, random mating, and no natural selection. There are 4 forces of evolution: mutation, gene flow, genetic drift, and natural selection. Natural selection for a polygenic trait changes the distribution of phenotypes. It may have a stabilizing, directional, or disruptive effect on the phenotype distribution. Lesson review questions. Recall. 1. Why are populations, rather than individuals, the units of evolution? 2. What is a gene pool? 3. Describe a Hardy-Weinberg equilibrium population. What conditions must it meet to remain in equilibrium? 4. Identify the 4 forces of evolution. 5. Why is mutation needed for evolution to occur, even though it usually has little effect on allele frequencies? 6. What is founder effect? Give an example. 7. Identify 3 types of natural selection for a polygenic trait. Apply concepts. 8. Assume that a population of 50 individuals has the following numbers of genotypes for a gene with 2 alleles, B and B. BB equals 30, BB equals 10, and BB equals 10. Calculate the frequencies of the 2 alleles in the population's gene pool. 9. Assume that a population is in Hardy-Weinberg equilibrium for a particular gene with 2 alleles, A and A. The frequency of A is P, and the frequency of A is Q. Because these are the only 2 alleles for this gene, P plus Q equals 1.0. If the frequency of homozygous recessive individuals, AA, is 0.04, what is the value of Q? Based on the value of Q, find P. Then use the values of P and Q to calculate the frequency of the heterozygote genotype, AA. Think critically. 10. Compare and contrast microevolution and macroevolution. How are the two related? 11. Explain why genetic drift is most likely to occur in a small population. Points to consider. Disruptive selection for a polygenic trait results in 2 overlapping phenotypes. Theoretically, disruptive selection could lead to 2 new species forming. How might this happen? Can you describe how it could occur? How else might one species diverge into 2?